We're going to go ahead and get started. Thanks everybody for joining us. Today is our second of our Timothy A. Johnson Medical Scholar Seminar Series. Just a brief mention about what this seminar series is. This was established, it's named in honor of our first Dean for Research here at VTCSOM, with the seminar series being focused on bringing in medical scholars who both practice medicine and contribute in a meaningful way to an area of clinical research. And so I'm just thrilled today to be able to introduce to you Dr. Sherita Hill-Golden, who is a professor of medicine at Johns Hopkins and also vice president and chief diversity officer at the School of Medicine there. Dr. Golden, though, is not unfamiliar with Virginia. She got her medical degree at UVA, and I believe she even spent a little bit of time here in Roanoke during those 4 years before VTCSOM. So we look forward to having her back someday when she can actually visit us at our campus. After she got her degree, though, she went on to begin her path at Johns Hopkins, doing her residency in internal medicine, a fellowship in endocrinology, and during her fellowship, she earned a master's of health science and epidemiology from the prestigious Bloomberg School of Public Health. And since then, she's been conducting NIH-funded diabetes research, so it's almost 20 years, with a focus on determining in a very holistic way the risk factors that surround the development of diabetes, including looking at things like stress and depression, as well as how that interplays with physiology and race and ethnicity. So she's published over 200 original research papers, invited reviews, monographs, at least 15 book chapters. But she hasn't just focused on the research, she's also focused on making sure that this continues and that she trains future physicians and researchers as director of an NIH training grant, looking at combining what she does best, clinical research and epidemiology. But then as you look through her CV, what I found interesting is that many of you know VTCSOM has recently decided to really focus on the concept of health systems science. And I noticed that Dr. Golden is not unfamiliar with this either. So she's, as a clinician, worked with her department and methodically evaluated the impact of the inpatient diabetes management service in the Department of Medicine, and worked with her team in a very systems-focused way to make fundamental changes in that particular service. So we could very much have her back to talk about health systems science maybe in a year or two. And of course, the other really fascinating thing about the publication record, as I look through everything that she's done, is not only do we have these very fundamental studies in diabetes epidemiology, but she's also contributing to our understanding of the impact of health disparities and also what it means to be and the specific challenges of being a woman in medicine as well as a Black woman in medicine. So she just, I don't know how she finds time to do all of this, but I'm very grateful that she's decided to spend a little bit of her very valuable time with us here today. She's going to present to us on her talk, which is Sugar and Salt, Assessing Adrenal Hormones and Diabetes Risk using Population Science. I invite all of you to ask questions using the Q&A button. You can put those in as they come up throughout the seminar, and I'll pop back on at the end to read those. Or if you prefer, you can raise your hand and I can unmute you if you're joining us via Zoom, and we can hear from you that way as well. Anyway, thank you so much, Dr. Golden, for joining us, and I'll hand it over to you now.
Thank you. Thank you for your generous introduction. I think that's the first time I've heard real clapping in weeks. So I'm going to share my screen. And there we are. So first, I do want to thank you for inviting me. And as Leslie mentioned, I'm so sorry I can't be with you in person because I've spent many memorable months down in the Roanoke Valley area. So, but maybe I can take you to some of my experiences there in the first part of my talk. What I was asked to do was to give a little bit of my career background, and I just met with a really impressive group of young trainees, your medical and graduate students, and talked about that a little bit, but just to sort of put that together, sort of how, I sort of arrived at looking at endocrine hormones and diabetes risk using population science, because when I was interested in endocrinology and pursuing a research career in endocrinology, it was very non-traditional for an endocrinologist to actually become an epidemiologist as well. So our objectives here are really, by the end, I hope you'll be able to identify 2 behavioral and 2 biological factors contributing to the depression diabetes association, to identify 3 ways to assess neuroendocrine activity in population-based studies, and to explain the biological mechanisms linking the renin-aldosterone system to type 2 diabetes. So if I think about where my journey started, it actually started in Virginia, believe it or not. So my maternal grandmother, who I'm pictured with there, grew up and was born in a place called Louisa County, Virginia, which is in central Virginia between Charlottesville and Richmond. And she ended up leaving Louisa County when she was 13 with about an 8th grade education, because there weren't very many opportunities for African Americans in the South. And just to put that into context, she was born in 1914. So she immigrated up to the Washington, D.C. area. And the first time that my husband, I found a record of her, was when she was 16 years old in Harford County, Maryland, which is about an hour north of where I currently live. And she was the nanny and the housekeeper for a white family in Harford County. The head of that household was in the military, so he got stationed from Harford County to Washington, D.C. And that's how my grandmother and my mother and my whole family ended up in the Washington, D.C. area. So both of my parents are native Washingtonians and met in high school. And so, but interestingly, my life story, I feel like in many ways did start in Virginia. So, there are going to be some very old pictures. I ask you, I'm glad I can't see you all so that you can refrain from laughing at some very old photos. But I actually developed my love for science and medicine when I was a young girl at Kettering Elementary School in Prince George's County, Maryland. And so it was my 4th grade teacher, Ms. Jan Taylor, was actually the first African-American teacher I had at that school. And, she saw something in me that I don't think I saw in myself. And, actually told my parents, I think she should be in accelerated classes. And so the next year in 5th grade, I was in Lillian Stroud's class. And that was where I actually fell in love with science and medicine. And so Mrs. Stroud taught us a unit on the human body as a part of our science class, and we only had to read 2 or 3 of those chapters. And I was so fascinated by it that I ended up reading the whole book, and decided at that point in 5th grade that I wanted to become a doctor. So Mrs. Taylor and Mrs. Stroud were very influential, as was Mrs. Margaret Bowles, who was my talented and gifted teacher. So here I'm pictured with them at my 6th grade graduation. And Mrs. Stroud actually lived to see me graduate from medical school. She is deceased now, but Mrs. Taylor and Mrs. Bowles are still alive and well. And then I went to Kettering Middle School, where we had to do a science fair project every year. So I made various parts of my parents' house a laboratory over time. This is actually in our family room by the steps in the basement. This was my 8th grade science fair project. But then I ended up going to Eleanor Roosevelt High School, which was a science and technology high school, where as a part of our senior research practicum, we were required to do a year-long research project. And I ended up doing mine at the Beltsville Agricultural Research Center, where I had done a summer internship for minority students. And I took the project I was working on there, looking at how ozone differentially impacted various strains of soybean crop. And I extended that over a year and was able to show that when we fumigated those various soybean strains with ozone, that certain ones were more protected than others. And the ones that were more protected had fewer stoma to let in the pollutant. And so this had implications, of course, for the growing of crops and how that might impact that. But I'm also picturing here Dr. Kent Mountford, because when I was in 9th grade, my very first science fair award-winning research project was with him. He is still alive as well, and we are still in touch with one another. But it was actually looking at how nutrients from detergents and pollutants were causing excessive algae blooms in the Potomac River and in the Chesapeake Bay where I lived. And so what we discovered is that that phosphate in particular really accelerated algae growth. And so he was able to use that data from my science fair project to then really set up guidelines about limiting phosphorus containing detergents from being poured into the waters around Maryland. So early on, I had this really this real passion and interest around science, and most of my exposure at that time was in the life sciences. So I ended up going to University of Maryland College Park for undergraduate and participated. I was telling the students about this earlier. I did 2 summers at the NIH. Again, these were summer research programs designed to encourage women and minorities to enter the field of science and medicine. And at that point, I was trying to decide, do I get a PhD because I really like scientific investigation and inquiry, or do I get an MD because I love the human body? And I was a bit torn. And so there was one summer where I worked in the lab at NIH during the day, and then 2 evenings a week I went and worked in the office of my grandmother's doctor, Dr. Richard Lilly, and I basically took blood pressures, EKGs. I was like a medical assistant, and I realized that what I really enjoyed was the human interaction with people. And back at that time, there was not a lot of the whole field of clinical research had not really evolved. This was like the mid to late 80s. So at that point, I decided I wasn't going to do research. I was going to go to medical school, and I was going to go to medical school to be a general pediatrician. And so this is my college graduation. I'll also point out that I was a first generation student. I was the first in my family to graduate from college. So that was a great thrill for my grandmother, who had made that long trip up to what was considered the North, even though Maryland's technically in the South still, when she was a young girl. So at the University of Virginia School of Medicine, and what was a blessing about going to medical school there is that it sort of was full circle for my family. So I remember when I told my grandmother that I was going to go to UVA for medical school, she actually was completely shocked. She's like, Virginia? But I tried to get you all out of Virginia. They weren't welcoming to me when I was a child. And, and the truth is, when she was a child, she was not welcome in the front door of the UVA hospital, and the wards were all segregated. But by the time I went there in 1990, there was a new hospital and the environment had completely changed. So this picture is she and I in the front of the new UVA hospital the day before I started medical school. So there were a lot of people that I met at UVA who were influential in my career. Dr. Apri was the Dean of Minority Student Affairs, who was very supportive of all of the students that were there. But the person who really changed my career trajectory was Dr. Eugene Barrett. So for the students, he's the one I was telling you about when we spoke at lunch, was an endocrinology faculty member that was my general internal medicine attending when I was a third-year medical student. And I was just so inspired by his passionate care of patients with diabetes, his ability to teach, and his sheer brilliance. And, he said to me, you ask a lot of questions that don't have answers. Have you ever considered a career in academic medicine? And he was the one that made me rethink this former passion I had around research and how I might bring that, into like where I was in medical school at the time. And I said, well, I did bench research and I didn't think that was for me. He said, well, there's clinical research and you can sort of combine that molecular background with clinical research. So I did my first clinical research project with him as a fourth-year medical student. And from that point on, I was sold. So I decided that I wanted to pursue a career in academic endocrinology, but with a focus on clinical research. So I ended up, and I just want to point out, and please don't laugh at the age of these photos, but this is when I was a third-year medical student, we did a part of our internal medicine rotation. I did at the Roanoke Memorial Hospital. I don't think it's called that anymore, so this is how old these photos are, but I did cardiology here at Roanoke Memorial Hospital. I did part of my pediatric rotation at the Community Hospital of Roanoke Valley, which I think has a different name now as well. And then I did my general surgery rotation at the Salem VA Hospital, which is not that far from Roanoke. So my parents actually drove down with my grandmother. I was mentioning to the students, the fall in Roanoke, if this is your first year there, is absolutely beautiful. So please go for a hike while you are there. And so that's a little bit of my background. And I told you a little bit about why diabetes, because Dr. Barrett had really influenced me, but there were 2 other reasons why I ended up pursuing a career in diabetes. And one of them is because the first patient I ever saw was a 35-year-old gentleman with type 1 diabetes. He was legally blind. He had end-stage renal disease and had undergone a kidney transplant. He'd had toe amputations from severe vascular disease, and he had such severe hypoglycemia unawareness that he'd gotten into 2 car accidents and actually had his medical license revoked. So he was completely disabled. And at the time, it was a growing public health epidemic. So this would have been 1991 to 1992. And it was clear that diabetes was disproportionately impacting minority and underserved populations. And so you might think, why would anybody want to go into the field? That all sounded, fairly discouraging. But when I was a 4th-year medical student in September of 1993, the results of the Diabetes Control and Complications trial was published. And it showed that if you tightly control glucose, you can prevent all the awful complications that I've seen in this very young patient of mine. So I knew I would have the tools to make a difference in the diabetes clinical care community. So by the time that I was graduating from the University of Virginia and on my way to Johns Hopkins for my residency, I knew I was interested in public health aspects of diabetes because this was affecting so many populations. I still had this love for molecular endocrinology, like I love endocrine physiology. And I also was interested in clinical research, but it was unclear to me how to really put that all together. So I would say that at Hopkins, I sort of ended up marrying all of my passions. So I will also say, because people often wonder, where does this fit in, that halfway through my residency, I met my future husband, Dr. Christopher Golden, who was a resident in pediatrics. So even though I originally planned to become a pediatrician, I always joke, well, at least I married one. So I still end up interacting with pediatrics. And he is currently an associate professor of pediatrics and the clerkship director for pediatrics for the rotation for our medical students and as a medical education scholar. But I also had another marriage while I was at Hopkins, and that is the environment where I was able to marry my love of epidemiology, molecular endocrinology, and diabetes. And so how that all came together is that in 2000, I attended my second American Diabetes Association meeting. And this was sort of the end of my endocrine fellowship. I was finishing my master's, and it was right before I joined the faculty. And I remember that the reason I show this picture of my son is because he was still a baby and was about 9 months old. And this was the first time I was going to leave him, even though in the able hands of my pediatrician husband, I was going to leave him and fly across the country. And so my mentor at Hopkins at the time, Dr. Fred Brancati, who was a wonderful diabetes epidemiologist, he and I ended up eating lunch at this Hard Rock Cafe in San Antonio, Texas, but it started to rain. And so we were stuck in this terrible thunderstorm and neither of us had umbrellas. So we were stuck in there for 2 hours and we just started chatting. And Fred said to me, what's all this literature I'm seeing about a link between depression and diabetes? Like, I don't understand, why would they be linked? And I said, well, actually, when we test patients for Cushing's syndrome or overactivity of the hypothalamic pituitary adrenal axis, the test we do is a dexamethasone suppression test. A normal person should be able to fully suppress their cortisol with a high dose of dexamethasone. I said, but patients with depression is where we often get a false positive. So the cortisol doesn't fully suppress. So they always have this slightly higher level of cortisol. And again, that was another light bulb moment for me, because I said, well, one reason people with depression can be at higher risk for diabetes is that they've got this overactivity of their cortisol axis, making them more insulin resistant and leading to diabetes. And so the moral of that lesson is that when you're eating a greasy hamburger in a thunderstorm, that you should listen to your scientific mentor. The two of you may come up with some really unique ideas together. So sort of shifting gears then, when we think about stress, some of you may be very familiar with this very famous painting by Edvard Munch called The Scream. And I have to say that during all of the things that come up frequently during COVID, especially when you're trying to organize hospital operations, I think many on our executive leader team have felt like this very often. And I'm sure for you all in medical school, when it's close to time for finals, you feel this way too. But the reality is stress is an important part of altering our physiology and contributing to chronic disease. And so at Johns Hopkins, we really, our Department of Medicine and our approach to medical training was really patterned by Sir William Osler, who was the first chairman of medicine at Johns Hopkins University. And he wrote a very famous book called Osler's Principles and Practice of Medicine that was published in 1892. And in it, he has a chapter on risk factors for type 2 diabetes. And if you look at the list here, these look like some of the traditional risk factors, like it runs in families. Ethnicity back then was actually white race and not Black race, because African Americans were still coming out of the post-Civil War era, and so we're still quite malnourished. So ethnicity and social class back then were those who were more wealthy, because they had access to more food and they would eat more, and so they were at higher risk. Adiposity, sedentary lifestyle, overindulgence, these are things we consider traditional risk factors. But Osler also pointed out that nervous strain and worry were also risk factors for diabetes. So he was actually already seeing, even back then, this connection between stress and risk for diabetes. And so what the literature was showing when I first started my career in 2000 and began thinking about this is that individuals with diabetes had about 2 times greater odds of having depression than those who did not have diabetes. And the lifetime prevalence of major depression was higher in individuals with diabetes compared to those without diabetes. And so we really tried to say, well, this could go in either direction. Those studies were cross-sectional. So it could be that having diabetes increases your risk for depression. And so one of my mentees, Dr. Brianna Mazouk, did a meta-analysis looking at studies that had looked at diabetes as a risk factor for development of depression. And when she pulled them, she found that there was about a 12% higher risk of developing depression if you had type 2 diabetes at baseline. And so the reason is because we know that people who have diabetes who develop new complications, particularly if they have multiple complications that result in any kind of disability, so visual impairment, sexual dysfunction, impairment in physical and cognitive functioning, can make individuals more prone to depression, particularly if that's all coupled with the lack of social support and passive as opposed to active coping skills. Those psychological demands can all lead to the development of depression. And so I was interested in exploring this question further using data from the multi-ethnic study of atherosclerosis, or the M.H.S study. And I'll just describe M.H.S briefly because I'll refer to it throughout this presentation. This was a multi-center longitudinal cohort study of the occurrence and correlates of subclinical cardiovascular disease and factors influencing its progression. And so participants were recruited from 6 centers around the US, as you can see, and it ended up being truly multi-ethnic. So at baseline, there were 6,000 men and women aged 45 to 85 years, and 40% were non-Hispanic white, but we had 10% Chinese, 30% African-American, and 20% Hispanic American. And none of them had a history of clinical cardiovascular disease. And so you might say, what does a cardiovascular epidemiology study have to do with diabetes and depression? Well, since depression and diabetes are both risk factors for cardiovascular disease, M.H.S had rich data on both. So we were able to look among individuals at baseline who did not have prevalent depression. We were able to follow them over the subsequent 2 years and see whether or not they developed diabetes. So at baseline, they were either categorized as having normal fasting glucose, impaired fasting glucose, untreated diabetes, or treated diabetes. And the treated diabetes typically were individuals with who were on medication and knew they had a diagnosis. The untreated were those who were previously undiagnosed. And then we had a follow-up measure of depression that we had at the 3rd visit. And so what we found is that compared to those with normal fasting glucose, those with treated diabetes had lower income and less education. They had reduced physical activity, and they also had a higher prevalence of diabetes complications. And ultimately, what we found is that when we just adjusted for age, race, sex, and M.H.S site, that individuals with treated diabetes had about a 54% higher risk of developing depressive symptoms than those who had normal glucose tolerance. And even after we adjusted for those differences in health behaviors, diabetes severity I just showed you, as well as differences in BMI and socioeconomic status, those with treated diabetes still had a 50% higher odds of developing depressive symptoms over time compared to those who did not, who had normal fasting glucose at baseline. So we had sort of shown that indeed having type 2 diabetes can increase your risk for depression. But what about the other way around, which was part of my original hypothesis? Depression can lead to the development of diabetes. And so again, in that same meta-analysis, Dr. Mazouk pulled studies that had been done at the time and showed that individuals with depression had about a 60% higher likelihood of developing type 2 diabetes over time. And so there are a lot of contributors to this. It can be that, again, there's a psychosocial burden of illness and lack of social support that can lead to obesity in the setting of depression. There are also differences in health behavior. So we know that individuals with depression are more likely to smoke. They're more likely to take in higher calories. Sometimes you can take in fewer calories with depression, but you can also tend to overindulge. They have reduced physical activity and less likely to adhere to the treatments. So that can increase their risk for obesity. In addition, some of our treatments for depression, certain serotonin reuptake inhibitors and tricyclics are associated with a higher risk of developing obesity. And then in addition, we know that there's, and this is where a lot of my focus has been, that alterations in the neuroendocrine hormones, so the higher cortisol and catecholamine levels and the enhanced inflammatory markers that are found in the setting of depression can lead to insulin resistance and ultimately to type 2 diabetes. So again, in MESA, we were interested in exploring this in the other direction. So this is the same cohort, except now at baseline, we have excluded individuals with prevalent diabetes, and we've defined them according to their depression status. And then we followed them for 2 subsequent visits to determine who developed type 2 diabetes. And so just like we had hypothesized that individuals who had greater depression symptoms, which in these tables are going to be shown on the right, they were less likely to exercise. So this is minutes per week of physical activity. They consumed more calories during the day. They were more likely to be current smokers, and smoking is an independent risk factor for diabetes. And they had slightly higher body mass index. In addition, those with depression had higher levels of interleukin-6 as well as C-reactive protein, so a slightly higher inflammatory profile. And so one of the questions is, did those things account for what we saw? So in our base model, if we only adjusted for age, sex, and race, those with depression had a 42% higher risk of developing type 2 diabetes. But if we accounted for those differences I showed you in BMI, lifestyle factors, and inflammatory markers, those with depression had about a 21% higher risk of developing type 2 diabetes. So even though no longer statistically significant, the risk didn't go to 1, which suggests that there's still some residual confounding and some factors that we may not have accounted for. So of course, my hypothesis is that one of the missing links in the studies that we had were assessment of neuroendocrine hormones, and really beg the question, are neuroendocrine hormones related to metabolic outcomes like diabetes and obesity? And if so, how do we assess them in population-based studies? And so this is sort of the sugar part of my talk, really focusing on the cortisol axis now. And so we know that stress affects hormonal factors that increase diabetes risk. So we're taught in endocrine physiology that any kind of stressor, whether it's physical, emotional, physiological, leads to an increased release of corticotropin releasing hormone from the hypothalamus. That stimulates the pituitary gland to release adrenocorticotropin hormone. And then that stimulates the adrenal gland to secrete cortisol. So if you're being chased by a bear, then you want this system completely activated because you want to mobilize your glucose that you need for energy. The activation of the HPA axis also simultaneously activates the sympathetic nervous system. So you have an increased release of adrenaline and norepinephrine. So again, all of this is to mobilize energy to enable you to escape from the bear. But then when you've escaped from the bear, then this is a tightly regulated feedback loop such that the excess cortisol feeds back to the pituitary as well as the hypothalamus and really shuts this whole response off. So, what we envision is that with certain stressors, this whole system may stay chronically activated at a low level. And the prototype of hypercortisolism from overactivity of the HPA axis, whether it's from a pituitary tumor or from an adrenal tumor, is Cushing syndrome. So as a clinician being trained in clinical endocrinology, we were taught the hallmark features of Cushing syndrome, which include acne, moon phases, sort of fat redistribution in the centripetal area, increased facial hair in women in particular, and a higher risk of developing things like diabetes, lipid disorders, and hypertension. So then, it's sort of like, how do we put this all together then? How do we link the brain and the pituitary to the pancreas? So what we hypothesize is that depression and mental stressors lead to chronic low-grade hyperactivity of the HPA axis. That leads to simultaneous activation of the sympathetic nervous system. So in addition to having increased cortisol, there's also increased levels of catecholamines. And it turns out that the sympathetic nervous system is also a potent activator of interleukin-6, which is an inflammatory marker that sets off a cytokine cascade. So we know that all of those hormones and inflammatory markers lead to insulin resistance. And ultimately that can lead to not only type 2 diabetes, but also cardiovascular disease, and may explain the link between the two of them. And if you look at the metabolic profile of depression, and obesity and type 2 diabetes, they actually share a common neuroendocrine profile. So in all three conditions, you can see elevated urinary free cortisol. You can also see increased adrenal gland volume in both depression and type 2 diabetes, suggesting that there is this tonic activation from the pituitary. In all three conditions, you can see an abnormal dexamethasone suppression test. And again, these are three common reasons for an abnormal test when screening for Cushing's. And then the cortisol awakening response, which I'll talk about in a second, which is that peak of cortisol that you see in the morning, and that indicates a healthy HPA axis, that peak response in the morning is blunted in depression and type 2 diabetes, as well as in some studies with obesity. So all three conditions share this neuroendocrine profile. And so one place that that has been able to look at this exclusively in a mouse model is looking at a transgenic mouse model that overexpresses 11-beta-hydroxysteroid dehydrogenase 1. And what this enzyme normally does is it regenerates active cortisol, which in a mouse is corticosterone, from the inactive cortisone. So this is a mouse model that overexpresses cortisol. And so what they were able to do was to knock out, they were able to sort of create this model where there is overexpression of 11-beta-HSD1. So this is when I become jealous of my basic science colleagues, because we can't knock things out in humans. The IRB doesn't like if we knock things out or add things in. But I think this is a really elegant model. Because if you look at the mice, the transgenic mice, which in each of these panels is shown on the right, you can see that they are more obese than the wild type mice. And then they subjected those mice to glucose, an intraperitoneal glucose tolerance test. And you can see here that the transgenic mice that overexpress the HSD1 have an enhanced glucose response to that glucose tolerance test compared to the wild type mice. In addition, when looking at the free fatty acids and the cortisol levels, free fatty acid levels, which are a marker of insulin resistance due to the fat turnover, free fatty acid levels in the circulation as well as in the portal circulation were higher in the transgenic mice than in the wild type mice. And then if you looked at the portal cortisol levels at the level of the liver, they are higher in the transgenic mice than they were in the wild type mice. So this model really supports our hypothesis. So then one of the challenges that we had was, how do you assess subclinical hypercortisolism in epidemiological and clinical studies? Because of the diurnal nature of the profile, it's hard to just draw one sample and then try to relate that to an outcome. So one of the technologies that emerged during this time was the ability to measure salivary cortisol. And what's great about salivary cortisol is you can assess it non-invasively. You can do timed collections to capture the diurnal variation. It can be collected in the free living state. And it also measures free as opposed to bound cortisol. So it's not impacted by conditions that impact the corticotropin binding globulin. It's stable for several days prior to processing, and it's strongly correlated with serum cortisol. So I ended up collaborating with Dr. Ana Diaz-Rue, who's currently the Dean of the School of Public Health at Drexel. And Ana had put together the multi-ethnic study of atherosclerosis stress ancillary study. So basically, when you have a large cohort study, you can take a subset of that cohort and do some additional, more detailed studies. And so what in that particular study, at that point, now at this point, MESA had gone out to 5 visits. So this is beyond the number of visits that I had access to when I first started the depression work. And you can see here that at visits 3 and 4, that there was an ancillary study where we collected salivary cortisol on 1,000 participants at the UCLA and Columbia sites. And so, and then in addition to that, there was a subsequent MESA Stress 2 follow-up study that I was the Hopkins site PI on, where we collected additional salivary cortisol samples at UCLA and Columbia again, but we also added Johns Hopkins as a 3rd site. So that enabled us then, between the 2 ancillary studies, to have about 600 individuals that attended both of these visits with collections of cortisol, and have enabled us to do some longitudinal studies. So what we ended up doing in collecting doing the protocol is we collected 6 samples throughout the day over 3 consecutive weekdays. And so the tubes were all labeled for the participants, and they collected their first sample at awakening. The second one is at 30 minutes after awakening. And then there were samples throughout the day to capture the cortisol diurnal profile. So at 10 a.m., at noon or before lunch, and at 6 p.m. or before dinner, as well as bedtime. And we collected samples before the meals because we all have a mild cortisol spike after eating. In MESA Stress 2, we actually collected 8 instead of 6 samples because we wanted to be able to characterize the diurnal curve, because we learned additional samples would be helpful from our first ancillary study. But overall, most of the time frame of sample collection was similar. We just collected 2 more samples in the afternoon to better characterize that curve. So this is what we were able to do is really sort of develop measures that accounted for each part of the diurnal cortisol profile. So we all have a baseline level of cortisol when we first wake up. So that was sort of what's characterized as A here. What's your baseline? But then the first 30 minutes of awakening, we have what's called the cortisol awakening response. So this is our peak cortisol rise for the day. So we could measure that with a slope. And then you have this sort of early decline here in the first 2.5 hours after awakening. And then there's a gradual decline throughout the rest of the day. And then you reach your bedtime or nadir cortisol somewhere around 11 p.m. or so. And so what we were able to do was statistically model this and basically deconstruct each of parts of this curve mathematically and look at how the slopes in the areas under the curve were related to diabetes, because that was the relationship that we were interested in examining. So I'm going to show you now a series of results that are the work of some of my really outstanding mentees. So Dr. Shivan Champanari was first interested in how the diurnal cortisol profile looked in the setting of type 2 diabetes. So this was a cross-sectional study to examine the association between salivary cortisol curve components and type 2 diabetes status in MESA stress. And so I'm just looking at the raw data. Patients who have treated diabetes are shown with the black dotted line, those with impaired fasting glucose with the blue line, and those with the red dotted line are those with normal glucose tolerance. So where we really saw some difference was here on the cortisol awakening response. And so you can see that the slope, that rise, is less robust in those with diabetes, and the decline is a bit less steep as well. And essentially what the results of this study showed is that compared to those who do not have diabetes, those with diabetes had a lower cortisol awakening response and a slower early cortisol decline. So they're suggesting that there's some abnormality in the axis. In addition to that, women with diabetes had higher total cortisol area under the curve than women without diabetes. So those women had more cortisol exposure. We did not see that same association in men. So there was a sex difference there. And I should note that these associations were independent of the presence of depression. But again, that was a cross-sectional study, and so we still weren't sure what the direction was. So it could have been that this blunted cortisol profile, may represent a neuroendocrine burnout in the setting of diabetes. The higher overall cortisol exposure that we saw in the women suggested, at least in that sex, that we were seeing the subclinical hyperactivity of the axis in diabetes. So either, sort of the burnout or the hyperactivity might be altered in the setting of diabetes and may be related to the pathophysiology. But again, we were stuck with the chicken and the egg. So then we began to explore these relationships more longitudinally. And so, for example, is diabetes at baseline and M.H.S stress associated with changes in the cortisol curve features over time? So this was the work of Dr. Elia Spanos, who's now an associate professor at the University of Maryland Medical System. And so he did the first longitudinal study with our data to determine if that diabetes status predicted change in the cortisol curve over time. And the two stress studies were separated by 6 years. And so the exposure at baseline again was diabetes status. And then we basically log transformed the cortisol curve features to look at all of the various components. And we use linear mixed effects models with cortisol sample values as the outcome variable and diabetes status as the exposure. And so what you can see here is that between M.H.S stress 1 and 2, the first study is shown here with a dark line in each of these graphs, and then M.H.S stress 2 is shown here with the red dotted line. You can see that whether you have diabetes or not, that the cortisol curve sort of shifts to the right, meaning that we all become a little more hypercortisolemic with age. And so the question is, independent of age and time, do we still see that same shift difference? Is it more so in diabetes than in those without diabetes? And to make a long story short, that there we really did not find a significant association between diabetes status and change in cortisol curve features when we made the adjustment. We did not have data on glycemic control and diabetes complications and other things, but it did not at least appear that having diabetes altered the cortisol curve dynamic significantly. So the other question is, what about in the other direction? How could cortisol maybe be related to hyperglycemia? So even though we didn't have measures of hemoglobin A1C at M.H.S stress 1, we did have it at M.H.S stress 2. So this was another cross-sectional study done by Dr. Joshua Joseph, and he's currently an assistant professor at Ohio State University. And so here, we examine the cross-sectional association of the diurnal cortisol curve features with glycemia, which we assessed by the hemoglobin A1C, but only could do this in M.H.S stress 2. So in this study, we looked at 850 men and women. Again, we used the same definitions of our cortisol curve features, and our outcome here was arbitrarily chosen as a percent change in hemoglobin A1C. And again, we use linear mixed effects models to model this and stratify by diabetes status. And interestingly, what we found is that in those individuals who had diabetes, that a flatter, earlier decline slope, so a positive slope means that it's declining less, and an overall slope that was declining less, were both associated with higher hemoglobin A1C. In addition, having a higher bedtime cortisol compared to a lower bedtime cortisol was associated with a higher hemoglobin A1C. And then having a higher total cortisol area under the curve was also associated with a higher hemoglobin A1C. So what that was telling us is that in the setting of diabetes itself, that hyperglycemia and sort of activation or higher exposure to cortisol seem to track with one another. And again, hard to sort out because this study was cross-sectional, but still indicating a relationship there. And so what we concluded was that among individuals with diabetes, there were cortisol curve parameters suggestive of higher HPA axis activity and dysfunction were associated with higher hemoglobin A1C. So either that meant that HPA axis dysfunction led to hyperglycemia in diabetes, that hyperglycemia led to the HPA axis dysfunction, or in fact, it was bidirectional. And so, these have been very complicated studies to nuance. And so because I mentioned that we have these 600 people, again, that have data at M.H.S stress 1 and M.H.S stress 2, we did one final analysis that was recently accepted to look at the longitudinal association of changes in the cortisol curve features with fasting glucose. So here we were looking to see, does the change in glucose lead to a change in cortisol features over time? And then that was the first analysis. And do changes in glucose then lead to, or do changes in cortisol curve features lead to changes in glucose? So we were really trying to look at this bidirectionally. And again, same features, your cortisol slope, your overall cortisol decline, cortisol awakening response in the area under the curve. And essentially what we found in that first analysis is that an increase in glucose is associated with an increase in the cortisol, the wake-up cortisol, an increase in the total cortisol area under the curve, and a decrease in the overall cortisol decline slope. So in other words, the change in fasting glucose, the higher your difference in your glucose is, then it looks like you have more evidence of hypercortisolism. In the second analysis, where we looked at, does the change in the overall cortisol slope lead to change in glucose, that the less your slope declines, then the higher your glucose. So these are complicated relationships, but basically what we found, where we found those differences was only in people with diabetes. So these data suggest that in the setting of diabetes, that there does appear to be this bidirectional relationship between glycemia and cortisol curve features. And so this is very, very complex and circular, like many endocrine hormonal systems are. But we know that, again, when there is a stressor, that there's sort of, typically there's tonic inhibition of the hypothalamus by cortisol, but in the setting of diabetes, as a physiological stressor, that does not seem to be the case, even independent of depression. So you get the increase in corticotropin releasing hormone, increases in ACTH, and that leads to higher cortisol levels. And what happens at the liver is that that results in increased gluconeogenesis, which can contribute to higher free fatty acids available because of the hypercortisolism to feed the gluconeogenesis. We know that free fatty acids can cause insulin resistance that impairs skeletal muscle uptake and can also impair beta cell function. And so then you get more hyperglycemia, which then results in more HPA axis dysfunction. So this is incredibly circular and bidirectional complex relationship. And so the one study that actually has shown that cortisol curve features predict diabetes is the Whitehall 2 study in the United Kingdom, which is an occupational study, cohort study, and they've assessed diurnal cortisol in a similar pattern to what we have throughout the day. And they have a larger cohort than we do. And they were able to show that higher bedtime cortisol, again, indicative of an abnormal HPA axis, predicted development of type 2 diabetes, and that having a flatter slope and a higher bedtime cortisol also predicted type 2 diabetes and impaired fasting glucose. So what they conclude is that alterations in the diurnal cortisol profile were predictive of glucose disturbances in diabetes. So this sounds like a lot of complex physiology, and you may really say, well, what does all of this mean? And so we know that in the setting of diabetes currently, we have a large armamentarium of therapy that you'll learn about when you do pharmacology to treat diabetes, but we still don't get to target and to goal in every patient. So this really suggests that we need to be continuing to look for novel approaches to primary prevention and treatment of diabetes to complement these existing measures. And so that, we hypothesize that modification of the neurohormonal response may provide a novel approach to primary prevention of type 2 diabetes. And there are some glucocorticoid receptor antagonists that are tissue specific that are in development that could be considered for this purpose. And my colleagues and I wrote an NIH grant to look at that, about 3 years ago or so, but I think they thought we were being a little ahead of our time, but we think that this is the next step in addressing this particular area. So ultimately, as we think about depression and hypercortisolism and all of those things, we really want to move our patients from looking like this, having an abnormal stress response, to looking more like this. That is ultimately, our goal. So in the last few minutes, I want to switch a bit and talk about salt. So it's probably apparent in this talk that I love hormones and I could talk about them all day. So I want to talk now about salt. So we know that, sorry, let me just take a break and cough for a second. Oh, excuse me. So if we think about the adrenal gland, I've spent a lot of time talking about cortisol and glucocorticoids. And so that really controls gluconeogenesis and our glucose metabolism. But we also have a set of hormones that control water balance and blood pressure. Excuse me. I'm also having allergies right now. So I have been involved in a few studies, as well as some of my other colleagues, that showed that Dr. Brancati, and early in his career, showed that hypertension was a strong predictor of type 2 diabetes in another cohort study. And then I'd also done a study showing that blood pressure in young adulthood predicted diabetes in middle age. And another team in our group showed that glycated hemoglobin was associated with a higher risk of hypertension. So this suggested some connection between blood pressure and diabetes risk. And so when I was first joined the faculty, I looked at whether or not blood pressure predicted the development of diabetes in a longitudinal cohort called the Johns Hopkins Precursor Study. So this is a study of about 1,500 white men, ages 20 to 29, at baseline, and they're all former Johns Hopkins medical students. So they enrolled in the study when they were medical students. And so what we defined as the exposure was blood pressure measured at baseline at age 20, and then it was self-reported during follow-up annually. And our outcome was incident diabetes. And so what we found is that these, and shown in the blue line, are individuals who did not develop diabetes. And then what's shown by the red line are those who subsequently developed diabetes. And we broke it out by systolic and diastolic blood pressure. And so what you can see is that starting at age 30, those who went on to develop diabetes had a significantly higher blood pressure trajectory than those who did not develop diabetes. That was true for both systolic and diastolic blood pressure. And in addition to that, the rate of change in blood pressure in those who went on to develop diabetes was significantly higher than the rate of change in those who did not. So this really led us to think about the renin-angiotensin-aldosterone system. And so again, just to review the hormonal physiology, we know that renin is released from the kidney, and it causes the conversion of angiotensinogen to angiotensin 1. Angiotensin 1 is converted to angiotensin 2 by a converting enzyme. And so in fact, one of our major approaches to controlling blood pressure, both in patients with diabetes and in other patients, is angiotensin converting enzyme inhibitors. This is what they target, because angiotensin leads to vasoconstriction. It also leads to efferent arterial constriction in the kidney, which leads to proximal tubule reabsorption, so increased fluid volume. And then angiotensin 2 also causes the secretion of aldosterone from the adrenal glands, and that increases collecting absorption as well. So anyway, this is the renin-angiotensin-aldosterone system is a really important hormonal system for maintaining vascular and water balance. So this is the work of two of my mentees. Dr. Joshua, you heard about earlier. He was the first one that said, the Jackson Heart Study has data on aldosterone, and there's a rationale and a physiological mechanism through which aldosterone increases risk for dysglycemia. So he partnered with Dr. Justin Tajui, who's now an assistant professor at Johns Hopkins in our division of endocrinology, to look at the role of the renin-angiotensin-aldosterone system in diabetes in African Americans using data from the Jackson Heart study. And so just to give you a bit of background, cell and rodent-based studies suggest that aldosterone excess may impair insulin secretion, insulin action, or both. And in the atherosclerosis risk and community study, several of our colleagues have shown that lower potassium, which is a potential marker of elevated aldosterone, was associated with higher diabetes risk. And then a lower serum potassium, again, a potential marker of higher aldosterone, and African Americans appear to explain 18% of their excess diabetes risk. So what Joshua and Justin sought out to do was to determine the relationship between aldosterone and renin, as well as insulin resistance, beta cell function, and prevalent and incident diabetes. So the Jackson Heart Study is a prospective study of the development and progression of cardiovascular disease in African Americans. At baseline, they recruited over 5,300 African American adults between the ages of 21 and 94 years old from a tri-county area of metropolitan Jackson, Mississippi. The enrollment began in 2002 to 2004, and there have been 2 subsequent in-person follow-up exams, and there's planned for a 4th one to launch soon. I should mention this is sort of modeled after the Framingham Heart Study, but in African Americans. And so this, again, study was longitudinal. And so at baseline, after excluding people without prevalent diabetes, there were about a little over 2,900 individuals who were non-diabetic at baseline. We measured their baseline renin and aldosterone levels, and then looked at incident diabetes through 12 years of follow-up. And I'll just show the incident diabetes data for the sake of time. But essentially, the aldosterone levels were log transformed because they weren't normally distributed. And they were broken down into quintiles. And so what you can see, this is sort of unadjusted models and then subsequently more adjusted models, but that individuals in the highest 2 quintiles had a significantly higher risk of developing type 2 diabetes over follow-up, despite all of our multivariable adjustment compared to those in the first quartile. When we looked at renin, the relationship wasn't quite as strong. And so we saw in a couple of our models that the presence of higher aldosterone in quintile 5 was associated with a higher risk of developing type 2 diabetes. But once we adjusted for body mass index, that association seemed to go away. So what we concluded is that among individuals without prevalent diabetes, higher aldosterone and renin were cross-sectionally associated with greater estimates of insulin resistance, which I didn't have time to show you that in the interest of time, and smaller compensatory increase in beta cell function, and then higher aldosterone and renin were longitudinally associated with incident diabetes over an average of about 8 years. And so renin angiotensin aldosterone system activation is a risk factor for diabetes in African Americans, and it may explain the high coexistence of diabetes and hypertension in this particular patient population. And so then if we look at the physiology, why might aldosterone be related? And I have to say, this is the fun part about working with younger physician scientists, is that I had never thought of aldosterone as a glucose regulating hormone, but it turns out that it has some of that capability. So that there are mineralocorticoid receptors for aldosterone, in the cells, and in fact, aldosterone and mineralocorticoids can actually cause activation of NADPH oxidase, which can result in the generation of more free radicals. And those free radicals can interfere with the PI3 kinase pathway that's really important in activating the insulin receptor and ensuring that the GLUT4 is transported to the surface to allow glucose uptake. So in fact, when we give an ACE inhibitor that blocks the development of the action of angiotensin. And when we give an angiotensin receptor blocker, we are in fact also potentially disrupting some of these pathways that could lead to dysglycemia through these intracellular mechanisms. And again, similarly, this is looking at insulin secretion. So this is looking at the beta cells. Again, aldosterone, mineralocorticoids can enhance NADPA oxidase to generate these free radicals that can lead to mitochondrial respiratory dysfunction that can ultimately impair insulin secretion. So again, using ACE inhibitors, angiotensin receptor blockers can really block the generation of aldosterone and block this process to prevent the impairment of insulin secretion. So again, these are the biological mechanisms that can explain what we're seeing in the epidemiology. So in closing, I just want to acknowledge my mentees in the Jackson Heart study and the other studies that I mentioned. There are a lot of overlap between the aldosterone work as well as the MESA work. The MESA stress studies was a very large team effort of several collaborators over a number of years, so I want to acknowledge them. I also want to acknowledge the funding agencies that supported this work. The MESA stress studies were supported through the National Heart, Lung, and Blood Institute. The early work I did looking at depression and determining how to identify and use salivary cortisol markers and all of those things were supported by my first Career Development Award from the Robert Wood Johnson Harold Amos program, as well as a K-23 through NIDDK. And then the work of the trainees that I pointed out, Dr. Champaneri, Dr. Spanankis, and Dr. Joseph, they were all supported by our Endocrine Clinical Research and Epidemiology T32 from NIDDK. I'm very grateful to my neuroendocrine mentor, Dr. Gary Wan. So I showed you Dr. Frederick Brancati earlier, who taught me everything I knew about epidemiology. And he unfortunately passed away from complications of ALS about 7 years ago, way too early at the age of 53. But Dr. Wan, when I did my K-23 award, really helped me understand how to measure endocrine hormones, how to measure cortisol, what the limitations were. And I was sharing with the students earlier that, recently he called me because he's about to retire. And he's like, Sherita, you still have like DNA in my freezer. Do we still need that? I said, I think we methylated everything that we could possibly measure. But I'm very grateful to Dr. Wan. His expertise is in neuroendocrinology of addiction. And he was sort of willing to take me on to help me really define the neuroendocrinology of metabolic disorders. So I'm forever grateful. And then, of course, I'm always grateful to my family. To my parents who have always been a source of inspiration and support and have allowed me to grow various kinds of algae, fungi, and bacteria in their kitchen during high school to sort of satisfy my scientific inquiry with my science fair projects. I'm very grateful to them for the many trips back and forth to Roanoke and Charlottesville. And then to my husband and my son, who are my two biggest fans. And so no, my son is not going to be a physician or a scientist. He is a senior studying journalism and African American studies at Northwestern. So my husband, I hope we didn't scare him away. So I thank you. I'll stop sharing my screen. I'm happy to take any questions.
Thanks so much. That was a fabulous talk. It covered so many different avenues, and it's inspired already at least 3 questions. So I'm going to read some of those questions out so that everybody can hear them, and then feel free to field them. So the first one is, could you talk more about how these issues leading to type 2 diabetes may disproportionately affect African Americans, especially based on our focus on the Jackson Heart study, since we as a group read the paper from that?
Yeah, so I will say that that's what inspired me initially to enter this line of work because, I, as a sort of like a stress researcher in a sense, I felt that, and I still believe that some of the stress from structural racism, discrimination, and other things were sort of contributing to adverse physiology in minority populations. And so what I was expecting to find, for example, in the MESA study, when we looked at depression and risk of diabetes and stratified by race, is that, it would be, the relationship would be stronger in African Americans and like our Hispanic population. Turns out depression is equally bad for everyone in that particular study. But if we think about this at a population level, and Dr. Diaz-Rue and her team actually looked at the differences in cortisol profile by race and ethnicity. You do see some alterations where, for example, in Latinos in our study, that curve is shifted slightly to the right, meaning that there's more hypercortisolism. And there have been studies showing that people who have depression and chronic stress have that same blunted, altered cortisol profile that's abnormal that you see in the setting of diabetes. So we don't have the cortisol profile data in the Jackson Heart study like we do in MESA, unfortunately. But I believe that any kind of work going forward, looking at the physiological contributors to disparities, if you have a study where, like in MESA, we have neighborhood census tract data, for example, and you can take a subset of that cohort and you can measure their cortisol, then you'd be able to make that link. I do think that we need to show that, some of those racial traumas are actually altering physiology.
Yes, very, very, very provocative results and exciting to base future studies on. Excellent. Another question, thanks for a great presentation. Has there been or are there plans underway to replicate the Jackson Heart study in other African-American populations?
That is an excellent question. Not to my knowledge right now because funds are so tight, but it needs to be done primarily because the Jackson Heart study, as fabulous as it is not representative of African-Americans in the US. It is a very, very different cohort. So I remember the first time I flew to Jackson, and it may have been like 16 or 17 years ago. I was just very struck by, the degree of poverty, and it's very different than like what we see in Virginia or Maryland. It's very different. So the population is not representative. They are a population with a very high degree of comorbidity. So if you remember, I said there were like 6,300 in enrollment. And when we excluded the people with prevalent diabetes, so we could do a longitudinal study, we were down to 2,900. I mean, we can't exclude people with prevalent hypertension. We only have 30% of the cohort left.
Wow.
So, it just goes to, there have been studies that do that, but you lose a lot of the cohort. So it is important, I believe, to replicate another African-American study that sort of takes, samples people from across the US more broadly, because it's great if you want to study the pathology, but it's not a reflection of the whole US Black population.
Great, very, very helpful. Is it known, another question from Kristen Howard, is it known if control of depression influences the risk for diabetes that it may confer?
Another great question. So what we do know is that most of the work has been done in people with depression and diabetes as a comorbidity, that if you treat the depression adequately, the glycemic control will improve. The abnormalities in the HPA axis improve, although the HPA axis never returns completely to normal, even in treated depression. And we're talking about, sort of more severe depression that requires pharmacologic therapy or things like that. There have not been any studies, to my knowledge, that show that if you take people with depression, treat them with antidepressants, that their risk of diabetes is lower compared to those who are not on antidepressants. And in fact, in the diabetes prevention outcome study, one of my colleagues showed that antidepressants actually were associated with a higher risk of diabetes. And part of that is because some of the antidepressants cause weight gain.
Right.
So we have had to be very careful in selecting even among classes. So it's not uncommon for atypical antipsychotics to be used as mood boosters to complement therapy with SSRIs. But some of those agents actually cause weight gain. So this is like the tension. So we have to make sure that we're choosing the ones that cause the least weight gain. And then as a part of cognitive behavior therapy, that's a part of treatment for depression. There's the whole increase in physical activity and the behavioral activation. So making sure that people are physically active too, but it's a catch-22. They feel better, but we still have the metabolic challenges.
Right, lots of complex interplays that you're having to predict and intervene with. A few more questions actually. So William Adams, ACE and ARB medications are growing treatments for hypertension and for heart failure. Are you aware of any data showing that those treatments may be more effective at preventing renal disease comorbidity than other types of heart failure treatment?
Yeah, that is a good question, and so they are protective. ACE inhibitors and ARBs are definitely renoprotective. And if you're thinking about like diabetes in particular, they're renoprotective and preventing nephropathy. So they're part of the first line treatment in patients with diabetes with hypertension who actually have evidence of microalbuminuria, for example. But ACE inhibitors are being used in hypertension, and they are in heart failure. And in patients with diabetes and heart failure, we definitely use the ACE inhibitors for those reasons.
Great. You've inspired a number of questions. I'm trying to quickly go here and see if I can pull out a few. Okay, so Miles Thomas, thank you, Dr. Golden, for talking with us today. Considering the data and findings that you and your teams have generated, where do you think priorities should be placed in terms of exploring novel interventions towards diabetes prevention and disparity mitigation?
I really think that, we've done almost 20 years of work sort of building the case that there is neuroendocrine alteration. So I think the next phase of work, and I'm hoping like my mentees will do some of this, because Dr. Wand and I wrote that grant, and I think they thought we were being so aggressive, and we were like, we're just old and tired now. So, but where I think the next line of work is that we know that there's probably a subpopulation of people that have sort of neuroendocrine activation as a part of their metabolic profile that makes them more insulin resistant. So those would be people with depression, but are there other groups? Who are those other groups that would fit that profile? Because then it's like, okay, we need to do like a clinical trial, and this is what we propose, where, we would give people a glucocorticoid receptor antagonist. So there are safe glucocorticoid receptor antagonists now that are used in the treatment of Cushing's syndrome. So really bad Cushing's syndrome. When you use the glucocorticoid receptor antagonist, the glucose levels come down. The problem is the blood pressure can go up because the cortisol levels go up. So you're blocking the glucocorticoid receptor. Cortisol can't bind, so it starts binding to everything else. So it'll bind to the mineralocorticoid receptors at high doses, and it can cause hypertension. But there are some currently in development, there are glucocorticoid receptor antagonists that are tissue specific. So that are specific to the liver, specific to the fat. So in that case, you could get that glucose sensitivity effect without the blood pressure and other mineralocorticoid spillover effects. And so I think and those are, there are some that are in clinical trials, I believe. So I think that when those are available, that would be the next line of work, is for people who have that sort of hypercortisolemic metabolic profile, identifying them and then doing a trial or physiologic study to see if you treat them, are there improvements in the glycemic control without elevating blood pressure and dropping potassium, and are there and what changes in the fat? Do we see actually changes in glucocorticoid receptor signaling at the adipose tissue level? Because that's where a lot of the activity is.
So sort of a precision medicine approach, both from looking at a population as well as the physiology and targeting it to tissue specific.
Exactly.
Well, I'm going to try to pull out one more question, and then probably we're not going to be able to get to all of these. Let's see. Okay, final question. Do you think that cultural differences play a role in the relationship between stress, depression, and diabetes?
So yes, and partly because different cultures sort of handle depression differently, and it can even manifest itself differently. And so, in many cultures, it's not appropriate to even acknowledge or express depression. And I think the stigma really has people suffering in silence. And it's not just that they're suffering in terms of their mental health, but their physical health too. I hope I've been able to convince you of that. And so, if I take my own community, the African-American community, I mean, for years, people sort of thought, oh, depression, just get over that. Like, we survived slavery. Like, you just need to go and get your stuff together, go to church, it's going to be fine. The reality is, correct, that is true, but take your SSRI and get your cognitive behavior therapy while you're doing that, And so, I know at our church, our pastor is like, we're going to talk about this issue, but we're going to call it Come Manage Your Stress Day. So that's what we did. And then we had that's what we called it. The psychiatrist talked about depression. And it was very helpful for many people. But, we felt like if we called it depression, no one would come. And so in a lot of minority communities in particular, there is so much stigma that people are not getting treated. And you really have to think about depression, like hypertension, like diabetes, like kidneys. If you have chronic kidney disease, we would not treat your chronic kidney disease. So if your brain is sick, why would we not treat that too? Right. And so, and I think this is the current climate we're living in. I think for a lot of minority communities, it is very, very stressful for a variety of reasons. I will just say, as an African-American woman who, doesn't matter that I'm a scientist and an endowed professor at Johns Hopkins, every time my son drives his car and leaves the house, I just can't sleep till I know he comes back. Like, that is a stressor. At some point down the line, that's going to end up impacting my physiology. So we have to think about the different environments that marginalized groups are existing in, the disabled community, the LGBTQ community. I mean, there are a whole host of marginalized groups that are sort of living with this chronic stress. Some of it is depression. They either won't acknowledge it or won't get it treated, but it's impacting their health.
Probably the most important piece to come out of any of this research. And thank you very much for laying the groundwork for connecting the physiology with these important chronic diseases and our setting right now. Thank you so much for spending time with us today, Dr. Golden. This has just been a fabulous talk and fascinating research, and I know our students have enjoyed it. Thank you.
Thank you for having us.